A brief intro. Oh, you're going to record this part too. Okay.
Yeah, is that okay?
No, I'm just teasing. It's fine. Just share this. I want to make sure it's. There we go. Okay. Well, thank you all for coming today. It is really an exciting day, and it's great to be here for TJ when he's getting ready to defend his PhD thesis. So, for those of you who've forgotten some about TJ's background, he came to us with a bachelor's in computer science from the University of Oregon. But since he's been here at Georgia Tech, he's been accumulating degrees. He got a master's degree here working with Eva Dyer, and then he got an MBA, which may seem like an odd choice, but some of you might remember that he has also been the co-founder of a company. So while he was, here trying to get degrees, trying to get his PhD work, he's also been getting a company off the ground. I think, his main role in that wound down a little while ago now, but that has been going on a lot of the time during his PhD career. And along with that, he's also been teaching courses for ECE and was nominated as the online TA of, won the online TA of the year in 2023, which goes to show you he's put a lot of time into his teaching and his education efforts as well. So he's been very, very busy. And so you might wonder, what does this very busy person look like? And this is the picture I took from our lab website. So I like this because I think it shows that even though he's very busy with all these important things, he can still have fun. And in fact, I think we've got quite a bit of evidence of that from the lab. TJ is always right in the middle of anything that's silly that's going on. So here's the Halloween photo. Here's our Christmas one. Now, I don't know, I've got to put Harry up against TJ to see who's got the craziest Christmas sweater, but TJ's definitely in the running. And then we've got the pirate swag. So a whole lot of the pirate swag, including this awesome giant flag in the lab, is something that TJ came in with. So he's really dressed up our research area. He's been around the world. We were in OHBM last year in Seoul, and it was great to see everybody and see, them in a new place. They've been out and about, and I hope you notice that TJ is still having fun and always makes these really interesting sartorial choices that I admire greatly. So just to end on a little more serious note, TJ is always in the middle of the lab, and he's always ready to support people, to help people with their projects, and just to hang out and talk to the people when they're having a tough time. And I really appreciate all the effort he's invested and all of the other students and in the lab itself. And it has been really great to have him in the lab. I'm looking forward to his defense today and being able to call him Dr. LaGrow afterwards. So TJ, I'm turning it over to you.
Great. Um, let's see. All right, is that coming through? Yes. Okay. All right. Well, thank you, Shella, for that wonderful introduction. Thank you, everyone, for attending my dissertation defense. During this talk, I ask that everybody please keep yourselves muted during the presentation. If a question does pop up, please feel free to write it in the chat or wait till the end. There will be a Q&A where everybody will have an open floor for any kind of questions. After, we will ask everybody to log off so that the committee and I can have a final closed discussion over the research. I'd like to first start off by thanking Dr. Vince Calhoun, Dr. Eric Schumacher, Dr. Matthew Block, and Dr. Sergey Pliss for serving on my committee, and Dr. Shella Kyle-Holtz and Jeffrey Davis for advising me for the better part of the last decade. My name is Theodore LaGrow, and the title of my PhD dissertation is Exploration of Spatiotemporal Dynamics in Neurodegenerative Functional Brain Networks. The objective of my PhD dissertation is to harness the capabilities of spatiotemporal analysis methods to understand the progression of Alzheimer's disease using functional brain dynamics. This morning, I will cover the background of my dissertation research, my dedicated aims, my contributions to the field, and some acknowledgments. My aims are broken down into three sections. The first, testing the stability and reliability of two computational methods for extracting patterns commonplace within the brain. Understanding these limits and benefits of this method, for my second aim, I apply these computational methods to a data set of longitudinal progression of Alzheimer's disease. Finally, I show initial work on the use of these projections of brain dynamics over disease development and further replicate and provide novel insights to the globally average fMRI signal, a controversial signal which may be more phase-locked to brain dynamics and diminished over disease development. By establishing methodological best practices and demonstrating the clinical utility of these methods, this work provides a framework for advancing functional neuroimaging biomarkers of Alzheimer's disease. Resting state functional magnetic resonance imaging, or RS fMRI, consists of scanning the brain when subjects are at rest, but not asleep. Within fundamental science and clinical settings, resting state fMRI has revolutionized our understanding of brain function and organization. The technique relies on the BOLD signal, which stands for blood oxygen level dependent and is an indirect measure of neural activity that relies on the magnetic properties of oxygenated blood in the brain. Over the past 30 years, through the principle of neurovascular coupling, we are able to use this measure to record large-scale brain activity over time. This approach has since become a valuable tool for investigating whole brain activity through the organization of functional connectivity within and between brain networks, and has led to important discoveries about the brain's intrinsic organization and alterations in various neurological and psychiatric disorders. Resting state fMRI studies have shown that the brain activity is not just localized, but there exist large-scale coordinated activities across the brain. One example of this is the resting state networks, which are networks of brain structures that have been consistently shown to exhibit similar activity at rest. Over the years, the canonical parcellations of the brainatome and Yo atlases have been used in many studies. To go further, when the data is organized into these canonically resting state brain networks, shown here at the top displaying scans of a cohort of concatenated healthy subjects, we see the empirical examples of repeated patterns of widespread coordination in the brain over time, as shown in the semi-repeating pulsations with sliding window and phase synchrony here. Resting state functional connectivity is typically obtained from this data by comparing the synchronous fluctuations of the BOLD signal between these different brain regions, which provides an effective measure for the strength of functional connections. The BOLD signal is central to this research as it provides a non-invasive clinical measure of the brain function and connectivity. Now, the lack of task makes resting state fMRI particularly attractive for patients who have difficulty with task instructions for evaluating cognitive decline. Resting state fMRI is non-invasive and relatively less expensive than other brain modalities, allowing for more frequent scans for patients with higher disease propensity on a longitudinal scale. Thus, the application of resting state fMRI in research and clinical settings have been a growing interest for the past 2 decades. Alzheimer's disease is a neurodegenerative disorder characterized by severe cognitive decline and decrease in neuronal connectivity and brain tissue. While many therapeutic approaches exist today to help slow Alzheimer's progression, medical communities still struggle with adequate prevention and treatment strategies. As it stands, Alzheimer's disease remains one of the largest threats to public health, impacting over 50 million people in the world, as reported by the World Health Organization in 2023. Alzheimer's disease is by far the most common type of dementia, estimated to account for 60 to 70% of all dementia cases. An estimated 6.5 million Americans aged 65 or older are living with Alzheimer's dementia today, with the number projected to grow to 13.8 million by 2060. The incidence of progression to later stages of Alzheimer's disease increases year over year. Moreover, the costs related to Alzheimer's disease are enormous, estimated to have reached $345 billion in 2023 in the US alone. The demographic composition of populations is skewed towards an increasingly proportion of the elderly, and relative costs are predicted to more than triple by the year 2050. Without medical breakthroughs to prevent, slow, or cure Alzheimer's disease, early detection is more important than ever. If dementia is discovered early in disease development, the number of Alzheimer's disease incidences can dampen by nearly 1/3 through rehabilitation exercises and medication. By identifying Alzheimer's early, we can ensure the growing population of elderly patients receive the care they need and provide fundamental knowledge to help slow and eventually prevent disease progression. Most investigators believe that Alzheimer's is a slowly evolving process that likely begins years to decades before clinical symptoms manifest. Due to the severity of effects caused by the disease, there is a strong interest in identifying individuals at an early stage. To clinically discern the progression of dementia, cognitive performance exams are administered in conjunction with subject-specific demographic information. Mild cognitive impairment, or MCI, represents an attempt to characterize subjects at an early clinical phase and has been the target for many clinical trials. In more recent years, while analyzing the progression of dementia of Alzheimer's type, or DAT, the most common type of dementia, several researchers have assessed subjects in specific cohorts based on the longitudinal progression. These cohorts will be the focus of the present research. In the first research group, Group 1, represents patients who do not change diagnosis during the window of study, usually many years, even a decade for some windows. The first two subgroups, normal control and mild cognitive impairment, represent subjects without DAT-related pathophysiological changes. These subjects do not have a diagnosis of dementia of Alzheimer's type during the window of study. The second research group, Group 2, represents patients who did progress in diagnosis during the window of study. Here, pre denotes the patients before a change in diagnosis, and post represents scans after the diagnosis. This allows us to look at the DAT spectrum that spans from a normal control to clinically asymptotic to severely impaired in a more granular perspective. To be able to use this method of resting state fMRI data for foundational science and clinical research, a number of pre-processing steps need to occur after the study collection. Pre-processing of fMRI data involves a series of steps to clean and normalize data to prepare it for subsequent analysis. The general pre-processing pipeline involves the following steps, many of which are study-dependent, where this list is not an exhaustive of everything that really could be done, but generally considered to be the bare minimum. Depending on the scan length and number of factors, a single scan can take anywhere from a half hour to an hour and a half, or even beyond, to properly pre-process everything, where each of these steps have a best practice or needs to be tuned. A standard data set, which consists of tens to hundreds of scans, can take months or longer to properly pre-process. Similarly, the time it takes to scan one volume of brain, or the repetition time, or TR, impacts the sampling of both the space and time of these abstracted patterns, potentially influencing the stability and interpretability of dynamic methods. Shorter repetition times provide higher temporal resolution, which may enhance sensitivity to fine-grained dynamics of the brain, while longer repetition times reduce the data acquisition demands but could alter the representation of the propagating patterns. One of the more controversial pre-processing steps is the globally averaged signal, or the global signal, where it is often used as a way to decrease noise in a data set. The most common way to do this is global signal regression. The rationale behind global signal regression is that the neural activity is localized, and thus any signal that is widely coherent is likely to come from non-neuronal processes such as small head motion or scanner noise, to name a few sources. Although global signal regression has shown to decrease various types of noises and artifacts, other research demonstrates widespread resting state signals, much like the resting state networks, and say that the global signal regression could be eliminating important neural information. New work continues to progress our understanding. However, this still remains an open question. Now, to better understand how these intrinsic networks interact over time, spatiotemporal patterns are used for the dynamic network analysis. These semi-consistent quasi-periodic waves of activity, known as quasi-periodic patterns, or QPP, were initially discovered by our lab in rats and have since been discovered in mice, humans, and non-human primates. These slow patterns of activity usually have a duration of 20 to 30 seconds in humans, occurring regularly throughout a scan. Here demonstrated is the algorithm illustration of QPP approach, followed by the demonstration of signals in human. This pattern is most characterized by the anti-correlated signal between the default mode network and the task-positive network, deferring more to restful and vigilance networks. QPPs contribute to a general whole brain functional connectivity of the brain, which can be altered in various diseases. Now, complementing QPPs, complex principal component analysis, or CPCA, provides an alternate framework for characterizing the spatiotemporal dynamics by leveraging both amplitude and phase information to extract coherent and structured patterns from resting state fMRI data. While QPP interactively refines a propagating template, CPCA decomposes signals into the complex domain, capturing phase coherent fluctuations that shape network interactions. A direct comparison of these methods were done in the Human Connectome Project dataset, where they revealed that QPPs and CPCA extract highly similar network dynamics, suggesting that they capture overlying features of intrinsic functional connectivity. Now, however, the use of the Human Connectome Project data, with its fast repetition time of 0.72 seconds and approximately 1 hour of total scan time per participant across 4 sessions per subject in a large corpus of participants, represents a high-quality data set and a pseudo upper bound on what is feasible for clinical applications. The extent to which these methods generalize to shorter scan durations, longer repetition times, common in standard fMRI practices, remains an open question. Addressing these gaps is critical for translating QPP and CPA analysis into applied research in more clinical context. To recap, I will be looking at the fidelity of QPPs and CPCA to assess the stability and reliability of these methods over scan duration, repetition time, or the time it takes each brain scan volume to be administered, the bandpass filter used for analysis. I will then use these methods in 2 groups of longitudinal Alzheimer's patients consisting of 7 distinct cohorts of disease progression. Finally, I will show initial results for utilizing these QPP-like templates to project into other data sets for disease differentiation and implications for the global signal, which likely is phase locked to specific brain dynamics. Now, for my aim 1, let's go into the fidelity of spatiotemporal patterns. Despite their promise, QPPs and CPCA remain highly sensitive to methodological choices, including scan duration, repetition time, and bandpass filtering. Variations in these parameters across studies can introduce discrepancies in detected patterns, limiting both reproducibility and generalizability. For example, longer scan times may enhance the stability of extracted patterns by capturing more recurrences of underlying network dynamics. However, they can introduce additional variability due to natural changes in brain dynamics occurring over longer timescales. In contrast, shorter scans may be more susceptible to noise and individual variability. These methodological differences underscore the challenge of achieving consistent findings across studies. Now, similarly, the repetition time, or TR, impacts the sampling of the spatiotemporal patterns, potentially influencing the stability of QPPs and CPCA. Faster repetition times provide higher temporal resolution, which may enhance the sensitivity to these finer scale dynamics, while longer repetition times reduce data acquisition demands, but could alter the representation of the propagating patterns. Notably, since the repetition time defines the optimized window length, it is inherently constrains the temporal resolution at which spatiotemporal patterns can be extracted and interpolated. Finally, bandpass filtering critically shapes the frequency contents of detected spatiotemporal patterns, with different filter choices enhancing distinct neural processes. To address these questions, this study systematically evaluates how scan duration, TR, and bandpass filtering influences the stability and fidelity of QPP and CPCA-derived patterns. Now, for this study, the resting state fMRI data were obtained from 5 independent data sets, totaling 227 participants across 350 scans. These data sets include the Human Connectome Project, a cohort of experienced video gamers, a data set trained by musicians from Northwestern, a mindfulness meditation study from Emory University, and a cognitive task-based data set from the Center for Brain Imaging at Georgia Tech. While these data sets include task-related fMRI scans, only the resting state fMRI data were used in the study to systematically evaluate the algorithms across a diverse set of data sets and acquisition parameters. Participation demographics are summarized in the table shown. An illustrated pipeline of this preprocessing is also shown. For this study, all data is global signal regressed. The first objective was to determine whether different data set methodologies would capture QPPs and CPCA. This figure compares QPP and CPCA-derived spatiotemporal patterns using the brain-at-home parcellation of 246 regions of interest. Zooming in, panel A shows the extracted waveform templates from both methods, demonstrating consistency in spatiotemporal patterns across the data sets. Note that the x-axis scale differs between QPP and CPCA waveforms. The QPP algorithm uses a window twice as long to account for the extended temporal structure, or long-tailedness, of the match filtered approach. However, the central waveform extracted by both methods scans the same core duration, around 20 to 30 seconds, allowing for valid comparison of temporal profiles. Panel B represents correlation matrices for the 246 brainatome ROIs, allowing for direct comparison between QPPs and CPCA-derived connectivity. The lower triangular section of each matrix represents the QPP-derived correlations, while the upper triangular section corresponds to CPCA-derived correlations, allowing for direct comparison across data sets. The difference matrix in panel C highlights methodological variations, with warm and cool colors indicating strong correlations in CPCA and QPPs respectively. Despite these differences, the overall patterns are highly similar, suggesting that both approaches robustly capture shared features of intrinsic functional organization. Similarly, this figure follows the same structure with the Yo network level, revealing that while both methods capture large-scale network interactions, the relative strengths in inter-network connectivity differ. To assess the fidelity of extracted patterns, we evaluated the correlation between the default mode network and other canonical Yo networks using the Pearson correlation. The default mode network is a core intrinsic network involved in memory, self-referential thought, and consciousness, with activity predominantly happening during wakeful rest and negatively correlated with external direct attention. Given its fundamental role in intrinsic brain activity, its correlation with other large-scale networks serves as a benchmark for assessing the stability and biological relevance of extracted patterns across different scans, durations, and analytical levels. Taking the network correlations of the default mode network, we can construct a polar plot shown here to better examine the differences between algorithms and data sets. We're looking at the graph, points more inward indicate an anti-correlation to the default mode network, or points towards the outside of the circle indicate stronger correlation to the default mode network. When examining these polar plots, we observe both consistencies and variations. While QPP and CPCA exhibit substantial overlap in the extracted network templates, variability is evident in the subcortical, frontal parietal, and task-positive networks across datasets and methods. These differences likely reflect the varying sensitivity of these methods to temporal resolution changes and their ability to capture transient network dynamics. Across all datasets, a consistent pattern emerges showing strong agreement in broad network structures, with divergence limited primarily to finer-grained inter-network coupling. To go further, we can systematically assess how scan duration impacts the stability of these connection patterns using the HCP and CABI-REST datasets. These datasets were chosen due to the large quantity of data available during and differing data characteristics. The HCP dataset includes 4 resting state fMRI sessions per study, each with a scan duration of approximately 15 minutes and a fast temporal resolution of 0.72 seconds, facilitating finer-grained temporal analysis. In contrast, the CABI-REST dataset includes up to 2 sessions per subject, each with a scan duration of approximately 7 minutes and a lower temporal resolution at 2.25 seconds, around 3 times greater than the Human Connectome project. This allows us to assess both the reduced scan duration and coarser temporal sampling effects, the reliability of the extracted spatiotemporal patterns. For individual subject analysis, we set 1 hour of total scan time per subject as the practical upper bound, reflecting real-world constraints for fMRI research. Longer sessions are often impractical due to the cost constraints, limited scanner availability, and participation fatigue. Furthermore, studies requiring repeated scans per subject face logistical barriers, particularly in clinical and developmental populations where extended scanning sessions may not be feasible. Given that HCP provides a maximum of 4 sessions per subject and CABI-REST provides a maximum of 2, we aim to assess whether meaningful spatiotemporal patterns can be extracted from a single scan or an accumulated scan time of up to 1 hour per subject. Now, for a group-level analysis, we considered scan durations exceeding 1 hour, reflecting common aggregation approaches in large-scale neuroimaging studies. Public datasets frequently compile tens to hundreds of fMRI data to enhance statistical power and generalizability. To ensure comparability across these methods, we set the upper bound for this analysis at 14.4 hours, aligning with the total available scan time for the CABI-REST dataset. This allows us to systematically examine how algorithmic performance scales with increased data availability. For a more intuitive demonstration, shown here, the x-axis is piecewise linear between the 2 scales. Now, to go further into some of the results, the subcortical area variability in the subcortical to default mode network correlation remains consistently high across scan lengths, indicating dynamic and heterogeneous contributions to the default mode network connectivity. This variability also reflects a lower signal-to-noise ratio in the subcortical regions, which are often less optimally captured by standard head coil configurations. Despite increased group-level averaging, subcortical correlation is not generally stable, suggesting a persistent influence in subject-level variability and acquisition-related limitations. Now, in contrast, the limbic network maintains a relatively stable correlation with the default mode network, exhibiting consistency across both algorithms and scan durations. Although this may seem unexpected if the signal-to-noise were the primary driver of variability, it suggests that the limbic default mode network coupling may be more reliable to both methodological and physiological sources of noise. Now, to look at the sensory cortex, the visual network and the somatomotor network remained consistently anti-correlated with the default mode network across both the individual feasibility and group level analysis. This pattern was observed both with QPPs and CPCA methods. However, the variability within these networks was lower for QPPs compared to CPCA, particularly for higher repetition data sets, such as the CABI rest. This suggests that QPPs may provide a more stable characterization of sensory networks, particularly in shorter scans with shorter TRs. For the association cortex, results indicate consistent trends across scan lengths and algorithms. The task-positive network remains highly variable, showing strong anti-correlation with the default mode network, where the dorsal attention network, or the DAN, exhibits greater stability across both methods. These findings highlight that QPP and CPCA may differ in how they capture association network dynamics. QPP-derived measures tend to show lower variability across scan lengths, which may facilitate interpretation at the individual level by yielding more stable templates. In contrast, the higher variability observed in CPCA-derived measures may reflect greater stability to intersubject differences, capturing individual heterogeneity in network expression. Furthermore, the standard deviation of default mode network correlations derived from CPCA remains consistently higher across both datasets, suggesting that CPCA is more susceptible to intersubject variability and may be less stable over shorter scan durations. In contrast, QPP-derived correlations exhibit lower variability, particularly in short scan lengths, implying a more stable and reproducible characterization of functional connectivity. These findings suggest that QPPs may be more suitable for individual level analysis, as it reduces variability for more reliable subject-specific characterization, whereas CPCA persistent variability may indicate greater suitability for group-level interference, where intersubject differences are averaged out over longer durations. Now, to evaluate how different repetition times affect QPPs and CPCA, we systematically resampled the repetition time, or TRs, across the data sets. The simplest method of synthetically increasing the TR for faster sample data sets was by selecting every second, third, or subsequent time point, effectively increasing the TR by integer multiples of the original value. For instance, here with HCP, or Human Connectome Project dataset, the native TR of 0.72 seconds was resampled to 1.44 seconds, 2.16 seconds, 2.88 seconds, and 3.60 seconds. Although there can be benefits in exploring longer TRs, we focused on resampling high temporal resolution data sets rather than interpolating lower resolution data, as the latter would introduce additional assumptions about the signal connectivity and could obscure native temporal structures of the QPP and CPCA components. To ensure independent comparisons across the TR values, each data set was divided into non-overlapping groups with independent subjects and scans, maintaining approximately equal total scan time within each group. This allows for controlled assessment of how TR influences QPP detection in CPCA-derived components across both fast and slowly sampled data sets. When examining HCP with approximately 3.5 hours of total scan time per group, both QPP and CPCA consistently preserve the dominant network dynamics, demonstrating that these methods remain robust for group-level analysis even as the TR increases. There is variability between the subcortical networks, frontal parietal networks, and task-positive networks here. This preservation in the HCP dataset, where despite resampling from less than a second to 3.6 seconds, the extracted QPP and CPCA templates remain structurally consistent across the 8 YO networks. To continue, looking at the video game dataset, following suit with consistently preserved dominant network dynamics. There were slight differences here between HCP between the sensory cortexes. Moving to the musicians data set, there is less data per group to account for the number of delineations. However, there are similar observations and consistency with the preserved data sets, especially within the comparison to the Human Connectome Project data set. Now, to finish, the meditators and CABI rest data offer longer repetition times to compare. Similar dynamics persist between these data sets and continued variability within the subcortical, frontal parietal, and task-positive networks. Now, to put it all together, this figure summarizes the last few figures and shows the default mode connectivity across data sets using both QPPs and CPCA. Violin and box plots show similar distributions of default mode network correlations across the resampled data sets with no significant difference between the method. This suggests that both approaches reliably estimate default mode network connectivity, even at varied TR, supporting their use in low-resolution fMRI data. Across datasets, the limbic network showed the strongest positive correlation with the default mode network, while the somatomotor and visual networks were consistently anti-correlated, except the video gamer dataset, which displays a more positive correlation, particularly in the ventral attention and the somatomotor networks. In the association cortexes, however, the dorsal attention network exhibited the strongest and most consistent anti-correlation with the default mode network, whereas the frontal parietal network showed a greater variability. The ventral attention network also maintained consistent anti-correlation. Now, notably, the task-positive network as a whole exhibited inconsistent correlation with the default mode network across datasets. These results suggest that the dorsal attention network may serve as a more suitable marker for task-positive dynamics in contrast to the broader, more variable TPN classification. Overall, default mode network connectivity patterns are preserved across datasets, TR conditions, affirming that both QPP and CPCA robustly capture intrinsic network dynamics in functional connectivity studies. Now, in resting state fMRI, bandpass filtering is commonplace used to isolate low-frequency bold fluctuations linked to spontaneous neural activity in most physiological rhythms. The most widely used range is the infraslow band, typically defined as 0.01 to 0.1 hertz. This range is standard in many pre-processing pipelines like CPAC and underpins much of what we know about functional connectivity. However, there is growing recognition that this infraslow range is not uniform and that studies often use slightly different definitions or cutoffs, leading to inconsistency across the field. Neurophysiological research has shown that this broader band can be meaningfully subdivided into narrow components, such as slow 5 and slow 4, each of which may capture different aspects of the brain. Slow 5 tends to reflect slower vasculature and metabolic rhythms, while slow 4 is directly tied to neural activity and dynamic connectivity. Now, to evaluate this even further, our study compares 4 bands, slow 5, slow 4, and the standard infraslow band, in a slightly extended band up to 0.15 hertz, which we refer to as band trunk. Including both the traditional and extended ranges helps us assess how higher infraslow frequencies may shape spatiotemporal patterns and influence variability we observed in the blood signal. Ultimately, this comparison helps clarify how different frequency definitions influence the detection of QPP-like patterns in network dynamics in resting state fMRI. Using HCP as a basis, we can first run QPPs over the 4 frequency bands for both QPPs and CPCA. Across all frequency bands, QPPs and CPCA analysis exhibited preserved network structures. However, distinct differences do emerge in how each band captures the temporal fluctuations in internetwork relationships. A notable distinction has observed in the slow 5 band, which captures periods of maximal interconnected synchronization where correlations peak or have reached their lowest values. In contrast, slow 4, infraslow, and the band trunk bandpass highlight change points in network dynamics, reflecting transitions in spontaneous activity rather than sustained synchrony. Interestingly, slow 5 closely resembles CPCA in emphasizing synchronized and interconnected network patterns, while slow 4 appears to be more uh, uh, sensitive to transitions between the states. Now, notably, slow 5 is the only band that excludes the 0.05 hertz peak commonly associated with QPPs at around 20 seconds per cycle. When evaluating the statistical relevance of this, Dunn's post hoc tests confirm that slow 5 differed significantly from the other bands, reinforcing its distinct temporal profile. We further quantified these differences by measuring the mean time lags between slow 5 and the other bands, revealing that consistent temporal offset. These results suggest that slow 5 captures unique oscillatory behavior aligned with peak correlation periods, distinguishing it from other bands more reflective of network transitions. This reinforces the alignment with CPCA-derived templates and its potential biomarker for coherent brain states. Additionally, the slow 5 window length can be analyzed to provide further insights into the temporal structure of these detected QPPs, where we can vary the window within the slow 5 band to see the observed QPP structure. Now, while shorter window lengths of 20 to 30 seconds capture inter-network synchronizations with previous findings, longer window lengths, 32 seconds, 36 seconds, and beyond, reveal that a distinct network transition phase occurs. This suggests that longer window lengths in the slow 5 are capable of detecting more granular changes in network organization and potentially capturing slower, large-scale reconfiguration in these brain dynamics for shorter lengths that may be overlooked. Notably, around 26 seconds, a shift was observed where QPP structures transitioned from capturing synchronized network fluctuations to detecting more complex spontaneous state shifts. Now, moving closer to the CPCA-derived templates, which they also emphasize moments of maximal synchronization rather than change points. These findings emphasize the importance of careful frequency band selection in functional neuroimaging studies, as different bands may highlight fundamentally different aspects of network dynamics. While slow 5 and CPCA provide a view of highly synchronized events, lower frequency bands such as slow 4 and the infraslow capture more gradual changes in connectivity states. To revisit the key questions pertaining to scan duration, repetition time selection, and bandpass filtering, we can draw some conclusions and recommendations based on the observed results. Longer scans improve QPP stability. Reliable QPP detection improves significantly beyond 10 minutes, especially for individual analysis. Shorter repetition times, less than 1 second, enhanced sensitivity, while multi-session data in interpolation can help mitigate variability in long TR scans. Shorter scans need group level support. Scans under 5 minutes produce unstable individual QPPs. Group averaging, pre-computed templates, or structural constraints can enhance the reliability. Subcortical patterns are less stable. Subcortical QPPs estimates are noisier, especially when with CPCA. Improving stability may require longer scans, larger samples, or even multimodal techniques like fMRI. Moving on to the TR selection, TRs, QPPs and CPCA preserve key network interactions like the default mode network and dorsal attention network interaction through and across TR values, supporting their use in varied acquisition protocols. They are clinically applicable. Longer TR data sets, such as Alzheimer's or Parkinson's open source data sets, still capture reliably large-scale dynamics, making QPPs and CPCA suitable for clinical studies in the future. They compensate with duration. Longer TRs required extended scan times to maintain resolution, and future work should explore adapted TRs or multi-resolution methods. Now, within the QPP versus CPA dynamics, QPPs track change points and dynamic transitions across bands, while CPCA captures coherent phase relationship. Frequency is still the key. Slow 5 versus slow 4. Slow 5 emphasizes recurrent synchronized patterns, while slow 4 captures the broader dynamic shifts and generalizes better across conditions. Similar patterns are extracted with a shift in phase. QPPs and CPCA offer complementary strengths. QPPs capture propagating network activity, while CPCA extracts coherent phase relationships and intrinsic connectivity. Using both methods together enhances the characterization of functional organization, and by redefining methodological approaches and ensuring robust analytical strategies, we can enhance the reliability of spatiotemporal pattern analysis in resting state fMRI. Moving to my second aim, I plan to show how these spatiotemporal analysis methods can be applied to longitudinal progression of Alzheimer's disease. Diagnosing Alzheimer's disease remains a major question and challenge, in part particularly because we lack non-invasive tools that can reliably track how the disease progresses in the brain. While fMRI shows promise, traditional static connectivity methods often miss dynamic, early-stage changes linking to cognitive decline. This study takes a different approach by examining QPPs alongside CPCA. By studying both in conjunction, we ask whether these spatiotemporal patterns can distinguish between stages of Alzheimer's disease, from cognitively normal subjects to mild cognitive impairment and full-blown dementia. We also investigate which brain networks show the greatest disruptions in these patterns as the disease progresses. To do this, we draw from two groups within the Alzheimer's Neuroimaging Initiative, or ADNI. The first includes individuals with stable diagnosis, those who remain cognitively normal, mildly impaired, or diagnosed with dementia over time. The second group, Group 2, includes individuals whose diagnosis changed within the study, progressing from normal cognition to impairment, from impairment to dementia. We label these scans from participants as either pre or post transition to track the changes over time. By combining stable and transitioning cohorts, this design allows us to examine both cross-sectional and longitudinal shift in brain network dynamics, providing a unique window in how QPPs and CPCA patterns evolve with Alzheimer's progression, offering insight into potential as early functional biomarkers. Shown here is the dataset, demographics, and the brain parcellation used. The NeuroMark version 2.2 Atlas offers a replicatable, data-driven framework for analyzing functional connectivity in resting state fMRI. NeuroMark is derived from large-scale group-level independent component analysis, ICA, to identify intrinsic network connectivities. NeuroMark systematically organizes functional brain networks into 7 primary domains and 14 subdomains, providing a multi-scale representation of brain function. To understand how the brain networks behave across stages of Alzheimer's disease, we started by identifying these repeated activity patterns for both QPPs and CPCA. Each template shows how different brain networks in the row changes over the time during the repeated cycle of activity. We then looked at how strongly different networks are connected during each of these patterns by measuring correlations within and between them. This gave us a snapshot of how synchronized or disconnected brain areas are, both within the same stage and across progression to see whether the network integrity breaks down. We compared each template's self-similarity to see how it compares with others. When these correlations drop, it may suggest a loss in coordination of the brain activity. We visualize the difference in this figure. We repeated this full analysis using both QPPs and CPCA, giving us 2 perspectives of how Alzheimer's affects the brain dynamics patterns over time. To test whether or not these changes are meaningful, not just random noise, we use statistical tests called the Krustal-Wallis test, which doesn't assume normality in the data. If this test shows a significant difference, we followed up with a post hoc analysis of Dunn's tests of difference with Bonferroni correction to pinpoint which groups were significantly different. Asterisks here show the statistical differences that were found. Here we see both group 1 and group 2, where the similarity and breakdown of disease coherence as the disease progresses. Notably, the CPCA template for group 2 for the unstable normal control post cohort exhibited poor separability of network dynamics when analyzed with CPCA. To extrapolate to see if there are other underlying dynamics, we can expand this analysis to higher orders of principal components. This presents the first 10 principal components along with their explained variance. The first component accounts for 29% of the total variance, while the second component accounts for around 26%, capturing a majority of the cumulative variance, yet showing minimal distinguishable signal across the networks. The lack of clear differentiation between the functional networks suggests that CPCA may not be effectively capturing meaningful structural differences within this cohort. And given these limitations, results from this cohort should be interpreted with caution, as they may not accurately reflect underlying functional network changes. Now, looking at both QPP and CPCA results for group 1, we see a consistent story of progression network disruptions as Alzheimer's disease advances, but each method highlights slightly different aspects of the progression. Starting with QPPs, early disruptions from normal cognitive to mild cognitive impairment appear in the paralimbic, subcortical, frontal, and default mode networks. As the disease progresses to DAT, changes spread more broadly, now involving the cerebellar, visual, and triple network domains, including the central executive and salience networks. By the mild cognitive to dementia transition, some of these disruptions persist, especially in the visual and triple network domains, while a few new ICNs emerge, indicating both stability and progression in functional breakdown. In contrast, CPCA captures a cleaner, domain-specific pattern. Early changes also emerge in the paralimbic, frontal, and default mode regions, matching QPPs and identifying early cognitive network disruptions. However, unlike QPPs, CPCA does not detect significant changes in the subcortical domain at any stage. As the disease advances, CPCA highlights growing involvement in the visual, cerebellar, and triple network domains, particularly the salience and central executive networks, while maintaining the same pattern of paralimbic and frontal dysfunction. The consistency of salience and executive network impairment across stages reinforces their role in AD-related cognitive decline. Overall, both methods point to progressive, domain-specific changes, but with some differences. QPP captures a more subcortical and temporally evolving transitions, while CPCA emphasizes stable, large-scale network disruptions, especially in higher order control networks. This complementary view helps strengthen the case for both using during the approach for study network level benchmarks in Alzheimer's progression. To contrast, in group 2, we examined subjects who transitioned between diagnosis stage, primarily normal to MCI and from MCI to dementia. QPP's results show a clear and progressive pattern of network disruption. Early changes begin in the cerebellar, visual, and triple network domains. As the disease progresses, additional alterations emerge in the subcortical, paralimbic, and sensio-motor networks. By later stages, higher cognition and basal ganglia regions become more involved, signaling a shift from cortical to subcortical breakdown. CPCA results similarly capture progressive dysfunction, especially in the cerebellar, visual, and triple network domains early on. Over time, there's a growing involvement in higher cognition in subcortical areas, though the paralimbic domains remain relatively unchanged in CPCA, suggesting method-specific sensitivity differences. A key finding across both methods is the sustained disruption of the triple network, particularly the default central executive and salience network through the disease course, underscoring their role in cognitive decline. Now, looking at the differences for group 1, both QPPs and CPCA detected key network disruptions, especially in paralimbic, frontal, and triple network domains. QPPs were most sensitive to subcortical and cerebellar changes, while CPCA highlighted broader visual and limbic disruptions. Both methods consistently identified frontal regions and have shown overlapping patterns in large-scale cognitive networks. Overall, QPPs capture deeper temporal structural changes, while CPCA reflects more disruption in cortical shifts. In group 2, both QPPs and CPCA capture key disruptions in cerebellar, visual, and higher cognition within the triple domain. However, QPPs were more sensitive to early and deep brain changes, consistently detecting subcortical, paralimbic, and sensio-motor disruptions than CPCA often missed. While CPCA highlighted broader cortical changes, highlighting the broader especially in the visual and frontal regions, it showed limited sensitivity to subcortical and salience network dysfunctions. Both networks picked up on default mode network changes, but QPP additionally revealed consistent disruptions in the salience and basal ganglia networks. Overall, QPPs appeared better suited for capturing transitional and deep network changes, while CPA still reflects more of a disrupted cortical reorganization. When comparing QPP and CPCA across key brain systems, we see that each method highlights different aspects of Alzheimer's related dysfunction. QPP consistently showed more sensitivity to these deep brain structures, especially disruptions in the hippocampus, thalamus, and cerebellum, and sensomotor networks, areas that CPCA largely missed due to its focus on broad variance patterns. In contrast, CPCA was more effective in capturing diffuse cortical breakdown, particularly in the visual system and the central executive networks. Notably, QPP also revealed stronger involvement with the paralimbic system and the salience network, which are critical for emotional regulation and cognitive control. So while CPCA emphasizes widespread cortical shifts, QPP provides a more detailed picture of early, localized dynamic changes, especially in subcortical and transitional regions. Together, they offer a complementary view of functional decline in Alzheimer's. In my final aim, I want to show preliminary results that warrant further research. I will show results for utilizing these QPP-like templates to project into other data sets for disease differentiation and the implications for phase-coupled disruptions to the global signal. Replicating results from rodent studies of QPPs from our lab, I projected QPP templates from healthy control data sets spanning the Alzheimer's spectrum to see how these patterns change with disease. What we found was a clear decline in QPP occurrence as disease severely increases, from normal cognition to MCI to dementia, suggesting that the brain's ability to maintain these structured patterns break down over time. This decline was consistent across both Human Connectome Project and ADNI data sets, and supports the idea that QPPs could serve as a dynamic biomarker for neurodegeneration. Because QPPs reflect an ongoing functional coordination, their loss may signal early disruptions before overt cognitive decline. However, some caution is needed. The HCP dataset is a younger demographic, so some decline may be reflected to the normal aging process rather than Alzheimer's alone. Looking ahead, future work should include age match in more diverse populations, integrating QPPs with traditional biomarkers like amyloid and tau, and explore longitudinal data to track changes over time. With further validation, QPPs could offer a powerful tool for early diagnosis and monitoring of Alzheimer's progression. On a separate note, co-activation patterns is a computational method to identify transient brain states from a single time frame in resting state fMRI. They reveal dynamic interactions between brain regions, unlike static connectivity, and have been linked to cognition, emotion, and clinical outcomes. CAPS help isolate brain networks like the default mode network and salience networks, and have potential for tracking disorders like Alzheimer's. But their interpretation is complicated by pre-processing choices such as seed region, thresholding, clustering, all of which can alter the results. CAPS can be sensitive to motion, global signal fluctuations, and physiological noise. Importantly, they may not always reflect causal network activity. Validation requires linking CAP transitions to known neural events or rhythms like the global signal. However, in a newer study looking at the global signal in rodents from the Alexander Gozzi lab, this new study demonstrates an initial overview of current replication and findings of the understanding of the functional brain states and their relationship to this global signal phase. This research centers around the study that happened in 2019, where the authors explored the dynamic patterns of brain activity in rats, finding that there is phase encodings or a phase-coupled interaction of functional brain states to the global signal. These results revealed distinct co-activation patterns occurring at specific phases of the global signal across the circular polar plots displayed here. What I have done is replicated these rodent findings in human studies, showing that the CAP transition align with the phase of the global signal, now demonstrated for the first time in humans using Human Connectome Project and the ADNI datasets for the normal clinical patients. Across different clustering choices, 2, 3, and 6, CAP transitions consistently occurred at distinct global signal phases. This global signal phase locking held true for both young adults and older ADNI participants, suggesting that it's a general feature of human brain dynamics. However, there are some limitations I need to note. The Human Connectome Project and ADNI dataset differ in age and temporal resolution, which limits the generalizability, so we don't yet know or understand the full biological mechanism behind the global phase coupling, whether it's more neural, vasculature, both, or something other. The next step for this research is to study the global phase coupling in a diverse set of clinical populations and across diseases. Additionally, I can also use multimodal techniques, more like EEG, fMRI, and longitudinal data to link caps to phase disruptions within the clinical decline. To summarize, the contributions of this dissertation, I have advanced spatiotemporal techniques for capturing dynamic brain activity, applied QPP and CPCA understanding to disease progression and brain signal phase coupling, and offered practical insights to methodological designs, Alzheimer's biomarkers, and rethinking how we should treat the global signal in analysis. In my first aim, I improved methodological rigor in spatiotemporal fMRI analysis. This work systematically evaluates how scan duration, TR, and frequency band selection impact the stability of spatiotemporal patterns in resting state fMRI. We found that longer scans improve QPP and CPCA reliability, especially in subcortical regions, and both methods remain robust across TRs. We also showed that frequency bands matter. Slow 5 enhances recurrent structures, while slow 4 better captures network transitions. More importantly, QPPs and CPCA offer complementary strengths. QPPs capture prolonged wave-like dynamics, while CPCA highlights coherent network phase relationships. Together, these tools support a richer framework for analyzing functional connectivity. In my second aim, I revealed that network disruptions across Alzheimer's progression. Using QPPs and CPCA, we mapped how these intrinsic networks deteriorate from healthy aging through mild cognitive impairment to dementia. QPPs were more susceptible to early transient disruptions in subcortical, cerebellar, and paralimbic regions, suggesting network instability even before full-blown symptoms emerge. CPCA, on the other hand, captures broader cortical degeneration, especially in visual and executive control. Both methods consistently detected triple network dysfunction, but focus on different components. These results highlight the need for multimodal dynamic biomarkers in AD research and suggest that QPPs and CPCA can help stratify patients by disease stage. In my third aim, I'd established global signal phase coupling as a functional marker. We showed that QPPs and CPCA reveal early signs of network disruptions in Alzheimer's. QPPs decline with disease severity, while CAP transitions remains phase locked to the global signal, even in older adults, highlighting potential early biomarkers and challenging assumptions about the global signal as noise. By integrating QPPs, CPCA, and global signal phase coupling, this work provides comprehensive perspective on large-scale functional brain organization. These methodological advancements hold significant implications for cognitive neuroscience, clinical diagnostics, and network neuroscience, offering new avenues for early disease detection, personal medication, and therapeutic intervention strategies. For this final section, I want to highlight the work and contributions to the field over the course of my graduate education. To start, listed here are all the relevant manuscripts pertaining to this research and tangential from collaboration within and between labs. For this work, I have 2 first author papers. Further listed are all the relevant conference abstracts and poster contributions over the years pertaining to this research. Additionally, I have completed several other projects while completing my master's degree, in addition to filing for a first inventor patent with my business that I co-founded. To highlight my contributions, I have authored 16 papers, including 4 first author contributions, 4 IEEE conference papers, 1 review papers, 2 software papers, and a myriad of method and analysis papers. I've contributed to 9 conference abstracts across 3 conferences, OHBM, EMBC, and ISBI, and I have 1 first inventor patent with my work with Insight Optics. During my time, I have earned several degrees, including a master's degree in electrical and computer engineering, an associate's degree in higher education, and a master's in business administration from the Scheller School of Business. While in grad school, I've had the opportunity to grow and learn through many departments through fellowships of leadership, education, teaching, and business. During grad school, I have earned a number of awards, including an NSF GFRFP honorable mention and several department and institutional teaching awards. Over the years, I've participated and won a number of competitions within the university, around the greater Atlanta area, around the United States, and through global competition, earning hundreds of thousands of dollars in non-diluted funds for the startup. In addition, I participated in service for the larger community, helping first-generation high school high schoolers with college application and essay writing throughout the springs and summers. My graduate education has allowed me to go to a diverse set of training programs. To help fund my education and pursue my passion for teaching, I have been a TA or an instructor in some capacity over the last 6 years. This spanned several departments and led me to even Berlin for a summer. I currently still teach graduate machine learning in the OMSCS department, where I have roughly 1,400 students per term, managing a staff of around 30 TAs. Throughout the education, training, awards, and teaching, I'm thankful for the experience to grow, develop, and contribute to the field. This dissertation embodies years of dedicated work, not only mine, but also those of countless individuals and contributors, often unseen, who have made this work possible. From the researchers whose findings laid the foundation to this study, to the participants who have volunteered their time, to the mentors who offered their guidance, and colleagues who have provided feedback, this work stands as a testament to a collective scientific endeavor. Their impact, though unrecognized by many, is deeply embedded in every stage of this research. I'm deeply thankful and grateful for everybody who has been a part of this experience. First and foremost, I extend my deepest gratitude to my advisors, Sheila Kyle-Holtz and Deb Davis. I'm consistently humbled by how incredible they are, both as scientists and as people. Their unwavering support over the years has really shaped my research and career in ways I can't fully articulate. I truly cannot thank them enough for everything they've done for me. I also want to express my appreciation to my committee members, Matthew Block, Sergey Pliss, Eric Schumacher, and Vince Calhoun. Each of you have left a profound impact on me throughout my graduate journey, and I deeply admire your work and strive to uphold the high standards you embody. I'm incredibly grateful to Georgia Tech, Emory, GSU, and the Trent Center in the greater Atlanta area for providing a dynamic environment to learn and grow during my 20s. Without Georgia Tech, I wouldn't have had the opportunities to attend advanced training programs, present my research domestically and internationally, and experience a community that's truly one of a kind. To my teaching mentors, Tammy McCoy and Kate Williams, they have played a critical role in shaping my teaching philosophy through many years working with the CETL department. I want to thank Wes Winans for allowing me to implement many of these teachings by guiding groups of incoming freshmen, developing and growing with a growth mindset through low stakes, high impact projects. Special thanks to David Joyner for allowing me to take over the machine learning class in the OMSCS department, which is a dream come true. This role continues to challenge me and grow as an educator. An important thanks goes to the administrators who have made my academic journey possible. Laura and Daniela, you are simply the best. I cannot begin to express how grateful I am for everything you have done for me over the years. To my lab members and collaborators, thank you all for the discussions, experiments, and camaraderie. From the endless hours in windowless offices to laughing at the absurdity of academic life, you have made this journey worthwhile. Machi, Harry, Lauren, Lisa, Michael, Nan, Xiaodi, Eric, Amrit, Wenju, Taylor, and Baipavi, I truly thank you. To my roommates over the years, Paul, Adriano, Chris, Philip, Phil, and Michael, thank you for sticking with me through the good times and the tough ones. You have been a source of support, laughter, and grounding through this process. With our weekly family meals, afternoon frisbee, board game nights, and finding fun ways to use excess recycling really got me through COVID. Those moments of joy and normalcy during such a challenging time meant the world to me, and I'm incredibly grateful to have shared the time with you. I'm nothing without my broader community. There are too many people to name, from senior grad mentors, whether they realize it or not, to peers who have made lasting impressions through small yet meaningful gestures. For those who are no longer with us, I hope to continue to honor and brighten our shared community. Both the Atlanta and Portland community in general area have been a huge part of my journey, and I want to honor everyone here with the images throughout the years. I will spare you all with reading out the names, but know that I cherish you all. To my family, Virginia, Renee, and Daniel, thank you for always being there since day one. I know my journey has taken many turns over the years, and I'm grateful for your love and support. I want to thank Virginia for allowing me and Jen to share your home while I finish my research over the last 2 years, as I'm very happy to have shared these last several years we got to live together. Thank you to my parents for teaching me to always strive for my dreams and always go beyond. To my extended family in Michigan, Karen, Mark, Teresa, Ashley, Ida, DJ, Gabby, your endless support has meant everything to me. To my family-in-law, Kathy, Alex, Logan, Matthew, BJ, and Mima, thank you for welcoming me into your family with love and support. I hope to make Pop Pop proud. However, this dissertation is dedicated to one of my biggest supporters throughout my life, Walter Ballou, or Papa. From attending my elementary school plays to showing up at every single one of my graduations, he's always been there cheering me on. Today, he is battling severe dementia, a disease that has deeply affected my family and one of my greatest fears. Papa, this work is for you, and I hope that in some small way, my research and journey honors the love and unwavering support you have given me. And finally, to my wife, Jen, the most important thank you of all. We met while we were in grad school, and through every late night, every challenge, and every moment of self doubt, you have been there. You have believed in me, supported me, and lifted me when I needed it the most. I love you endlessly, and for all the sacrifice you made, I hope to spend the rest of our lives repaying you with the love and appreciation you deserve. I sincerely appreciate everybody listening to my defense this early in the morning. Thank you all. And with that, I will open up the floor to questions.
Great job, TJ. If people want to put questions in the chat, I'd be happy to read those out if anyone has some, or if the community would like to unmute, that is also okay. Oh, I see there's one in the Q&A. Oh, it just says, so proud of you, TJ. Any questions from the general audience? Well, if not, I would ask everyone who's not on the committee to sign off so that we can go on to the next portion of the presentation.